Okay, so yeah, 15.1, we're looking at what's called an operon. And, well, broader speaking, like all of chapter 15 is looking at gene expression. And this is kind of an interesting example. So, like, this fish, it has one eye, but this eye, the top portion of the eye needs to be able to see, like, in a medium of air, and the bottom portion needs to be able to see in the medium of water. But, like, all of the cells in this fish's body all have the same genes. So the question is, how does this top portion of the eye be able to, like, be adapted to seeing in the air, and the bottom portion the water? And the answer is gene expression. The top portion of the lens of this fish will express, when I say express genes, I'm saying use genes. Use those genes to make proteins. The top portion of the fish's eyes are going to use different genes at different times, at different amounts, so they can see in the air. And the bottom portion of the eye will use different genes at different times to see in the water. Okay? So kind of getting this idea of, like, there are different kinds of cells in the body. Each cell type will doesn't have, like, new or different DNA from other cells. It's just it uses the DNA in different ways. Okay? All right. Whether it's a prokaryotic cell like a bacteria or a eukaryotic cell like that fish or you, all cells will alter their gene expression depending on environmental conditions. Because think about it. Whether or not, like, what kind of nutrient, like, think of like a bear in hibernation, right? The kinds of genes you're going to need to use to survive in hibernation, like how often or all that use certain genes, is going to differ versus if you're a bear, you know, chasing after your prey or something. And all of that, again, just comes back to gene expression. So environmental conditions and gene expression. We talk about, like, homeostasis. All of those kind of words can kind of be lumped together in a way. And thinking about evolution, like, you're thinking about, like, well, how is like some of this stuff, like, evolved? The idea would be, like, all of life has evolved. Nature has selected for organisms that can change how often they use or don't use certain genes. Like, that's like if you're good at changing gene expression, you're an organism that good at changing gene expression to meet environmental conditions, you're more likely to survive longer. And if you survive longer, you statistically will have more offspring. Okay. And then when we went over metabolism, we talked about, like, feedback loops, that, like, the ending product of one pathway could feed back and inhibit, like, the first thing in the pathway. A similar thing is going to happen in gene regulation, where you have feedback inhibition, that the product will feed back and inhibit the first thing in the pathway. And I'll show you a picture, you know, going over that in a little bit more detail. But yeah, we're going to be focusing on what's called an operon. An operon is a collection of genes that work together. So, operon, basically a bunch of genes that work together. And not only work together, they're also located together. Which makes sense, right? Think of the science hallway. Like, all of the science classrooms are all in one hallway because it's just more efficient to do things that way. It's more efficient to have all of the gas lines for chem classes be in the same part of the building, or you know, all the microscopes for bio classes be in the same part of the building, so people can share resources. A similar thing, and this is only bacteria. I want to emphasize that only bacteria do this. Eukaryotic cells will do it a little bit differently. How we can control genes that work together. Bacteria, they will just cluster genes that work together into these things called operons. All right, so let's get into some of the details of it. So looking at feedback inhibition, this left-hand part of the picture is something we learned about, way back, like, I think before winter break, you know, where, all right, so, you know, usually things we need to produce in, like, an anabolic pathway. Anabolic is where you're going to produce something, where you need enzymes to make that thing. And the ending point, so here we're making tryptophan, or the bacteria is making tryptophan. Tryptophan will feed back and inhibit the first part, the first enzyme in that pathway, if you have enough tryptophan. And that makes sense, right? Because, you know, we're all limited in the amount of calories, the amount of energy we have in a day, right? We don't want to waste resources. So if you're a bacteria or us, you don't want to make more tryptophan than you need. So it makes sense that, like, when you make tryptophan, tryptophan will go back and start shutting off like the sink, if you will, so you don't keep wasting resources. How that relates to gene expression is enzymes. Enzymes are proteins. The information for those enzymes are encoded into genes. So this is kind of adding in this bit of the biology that we've learned about in this unit, where all of these enzymes are controlled by sometimes multiple genes. Like these two genes, like notice how there's 2 parts to this enzyme. This would represent 2 different polypeptides, each polypeptide coming from a different gene, would be the idea. Anyways, tryptophan would shut off the expression, the use of those genes. Okay? So, makes some sense, right? So in other words, genes have an on-off switch. Okay? And here's like another kind of more formal definition for an operon. A single promoter. A promoter, that's where the RNA polymerase binds. RNA polymerase binds and starts transcription. Is that the promoter? Okay. So an operon, a bunch of genes are controlled by the same promoter. And think about why that makes sense, right? Because if all of those genes, like if I go back to here, if all of these genes are needed to make tryptophan, it would make sense that like if I'm going to turn on one gene, I want to turn on all of them. Think of it this way. Like there are 9, 12, 15 lights in here. All of those lights are controlled by one switch. Like when I come and I flip on that one switch. Can you imagine how annoying it would be if I would have to like go around my room and turn on individual lights for each of the, like it'd be dumb. It's like in a way it could be nice if I could have that level of control, but like I always use those same lights. And so, it's more efficient to have one switch controlling all those. In a similar way, it's more efficient to have one switch for a bacteria that controls all those genes. Okay? So again, there's a certain amount of common sense to how the operon will work and efficiency. Okay? All right, so let me show you a picture of how all this works. And I know this seems like a lot, but you can understand this. You don't have to blindly memorize it. This can make sense. So let's just kind of go through it one at a time. Starting with the left-hand part, there is something called a regulatory gene. And this regulatory gene, it has a promoter. It's where the RNA polymerase would bind. This regulatory gene, they call it TRP. The TRP stands for tryptophan. So that's the amino acid we were talking about earlier. So notice how all of these genes have TRP. Here, the R in tryptophan stands for regulatory. Okay? This will make transcription of this gene makes the mRNA for that protein, and then the actual protein is a repressor protein. A repressor represses things. If you're a repressor, if you repress things, you turn things off. You're like a hater. This protein turns off. Well, what does it turn off? It turns off this operon. Operon's a collection of genes. So all of these genes, TRPE, TRPD, TRPC, TRPB, TRPA, all of these polypeptides, all these proteins, these make up enzymes that we need for tryptophan synthesis. I'll keep the same abbreviation, TRP synthesis. Okay? Now, if you're a bacteria and you need tryptophan, this operon is on. Okay? We're making the mRNA because we need to make tryptophan. Okay? And this is what you see here, right? If the bacteria needs tryptophan, RNA polymerase can bind to its promoter, and it can go through and it can make all of these the enzymes you need to make tryptophan. Now, notice here there's something called an operator. So the repressor, if you look at this little puzzle piece and then this little puzzle piece, this can fit into the operator, but only if it's in the active form. So it only binds there if it's in the active form. Right now it's in the inactive form. So the question is, well, how do we make it become active? Well, if I go to the next slide, what makes that repressor protein become active is when you have tryptophan, which tryptophan functions as a co-repressor, meaning When there's enough tryptophan in the cell, we've made enough of it, we will now activate this co-repressor tryptophan, bind to the repressor, and then that repressor will bind to the operator, and then we shut off all of those enzymes that we need to make tryptophan synthesis. It looks like there's a lot going on, but it makes common sense, right? If I don't need tryptophan, turn off the faucet. If you got enough water, turn off the faucet. Okay? We make enough tryptophan. Tryptophan is the same thing as what's going on here. You make enough tryptophan, it binds, and it shuts off all these genes. That's all this is trying to show you here, is that we're shutting off those genes. It makes sense? I think it's really cool. Like, I think it's kind of wild that it's that elegantly simple here. Now, little subtle detail. What this is trying to show is that the repressor gene actually is located much further along on that circular bacterial chromosome than all the genes that we need to make tryptophan. So technically, this wouldn't be part of like the operon. If I go back here, the operon is just referring to these genes that are clustered together that make the tryptophan. Okay. Make sure I said everything. I think we're good here. So this is just in words, everything I was saying. I'll look through it, make sure we're all good here. Co-repressor, if like you're cooperating with somebody, you're working together. That's why tryptophan is a co-repressor. It works with the repressor. And we call this a repressible operon. So repressible operon. So TRP operon. And I'll get more into what that means here in a little bit, but just kind of keeping that in mind. This would be an example of a repressible operon. So in a repressible operon, default equals on. Meaning default is the genes are on. We're using the genes to make the tryptophan. Okay. So moving to talking about repressible operon, I now want to talk about what is called an inducible operon, which is basically like the opposite idea of a repressible operon. Okay, repressible, you need a repressor. So repressor on operator, and that turns off transcription. Okay. Now, inducible operon here. This is going to be the lac operon, which I'll show you a picture of. That's the example. Here, default is off. So in an inducible operon, our default is it's off. Meaning we're not using the genes there. Okay? In other words, we don't have a repressor, we have something called an inducer. So if a repressor turns off transcription, what do you think an inducer does? It turns it on. It induces it. It turns on transcription. Okay. So let's look at the details of it. So here we're looking at lactose. If you have like issues with lactose, like you're very familiar with, lactose is like a milk product. Lactose is basically a disaccharide sugar where like we have an enzyme called lactase that will break it down. Some people, like I personally, have some issue with my lactase where lactose can cause some digestive issues. Anyways, if you're a bacteria, you can use lactose to get energy out of it. Now you need to make the enzymes to break down that lactose. So here's the deal: if there's no lactose, why would you make the enzymes to break down lactose? Like, if you're not driving anywhere, like, notice how all the cars out here are off. That's because you're not driving them. So, inducible operon, like the lac operon, its default is off because Why would it be on? That's a waste of energy, a waste of resources to keep these genes idling to keep them on. You only want to turn them on whenever you have lactose present. So in other words, here the repressor is default. The repressor, its default is it's bonded. So repressor. Here, I'll edit this. Default off, and that's because the repressor is bound. Okay, that's why the default is off, because your repressor is bound. Here, default is on because repressor is unbound. Okay? So I think it makes some common sense here. Now what's interesting is notice how there's no little squiggly line. The regulatory gene for the lac operon is actually present like right around the same stretch of the DNA. Not super important, but kind of an interesting little detail. But here, because you have the repressor there, RNA polymerase can't make those enzymes we need to break down the lactose. Okay, so now then, let's say you're a bacteria and like I put a bunch of lactose in the medium that the bacteria is growing in. Well, now, here is that lactose. Now, technically, there's something called allolactose. Allolactose, they call it an isomer of lactose. Basically, it's same chemical formula, just different rearrangements of the atoms of lactose. Think of it like lactose, okay? Functionally, it's like lactose. It's a sign that lactose is around the bacteria. This allolactose, it is a inducer. Said earlier, inducers turn things on. This inducer, how it turns it on is indirectly. So it's an inducer indirectly. What I mean by that is, it isn't directly causing us to like make RNA polymerase to make these, like this allolactose isn't directly making these enzymes. It indirectly affects it by binding to the repressor. The repressor was here, right? The repressor was being a hater, binding to the operator. Little vocab, that's the operator. So repressor was there, but because lactose is in the medium, lactose binds to the repressor, inactivating it. Now RNA polymerase can bind to, what's this called? The promoter. It's a promoter because it promotes things. Okay, binds to the promoter, and then now it can go along and go through the operon and make all of those genes, use all those genes, transcribe all those genes you need to make all those enzymes. Notice directionality, 5 prime, 3 prime. Notice how each of these enzymes like is coming from one mRNA strip, and there's a start codon, stop codon. That would go with, you don't need to know these enzyme names, that's not important, but what is important is understanding like they all have their own start and stop codon. That is an important detail to know. All right, any questions about this? Okay. Again, basic idea. Don't get lost in the details. Basic idea makes some sense, right? You only want to make the enzymes for lactose when you have lactose. Okay. And again, that is an inducible operon. Now, both of these examples here, these are part of what we call negative gene regulation. And I'll explain what positive gene regulation is. Anyways, just keep that in mind for right now. Whenever the enzymes in inducible operon, they're called inducible enzymes. Inducible enzymes are enzymes that can be induced. They can be turned on, whereas for the repressible operon, the enzymes are repressible. Repressible, meaning they can be repressed. Makes some common sense there. Another important detail, distinguishing repressible and inducible. Repressible operons, we are going to use these for anabolic pathways. Inducible, we use these for catabolic pathways. And again, you don't really have to memorize this. Try to make this make sense to you. Tryptophan was something we built. Okay? It makes sense when you build things. When you have enough of what you build, you want to stop building. Anabolic, that was a fancy like metabolism word for saying you take you go from monomer to polymer, right? You're building things up. Catabolic, that was the opposite. That's where you go from polymer down to monomer. Okay? So any pathway, any like sets of genes that are like involved in anabolism, building things up, we are going to control those in a repressible way, where they can feed back and inhibit each other. Whereas anything that's a catabolic pathway, so here lactose is a polymer, right? It's a bigger sugar. We break that polymer down into its individual monomers. Okay, so it makes sense that we only want to turn that pathway on when it's present. Okay. Okay. Now then, looking at kind of another interesting example here. So, if you're a bacteria, they would like to use glucose. If you're a bacteria, you would rather use glucose. We talked about glucose was used in like cellular respiration. That's the most efficient form of energy if you're a bacteria to use. Now, let's say, we talked about how they can use lactose to get energy, but like, let's say, like, let me give you a scenario here. There is lactose, so lactose is present, but glucose is scarce. So, building upon what we learned about the lac operant, if you have lactose present, allolactose turns off the repressor, and we can make those genes we need to break down lactose. But there's another variable we have to throw in here, because if you're a bacteria, you would rather use glucose. Now, if you don't have glucose, you want to really ramp up. This is getting into what we call positive gene regulation. You really want to ramp up production of your lactose genes. How you ramp those up is the signal when glucose is low. So low glucose means you have high CAMP. We learned about CAMP when we did like some cell signaling stuff. CAMP is cyclic AMP. So cyclic AMP, here's CAMP. If I have low glucose levels, CAMP levels build up inside the bacteria. Those CAMP levels will inactivate something called CRP. This is CAMP receptor protein. So the name kind of goes along with what it does. CAMP binds to CRP. When CRP is active, this will promote, so active CRP will increase GE, increase gene expression of the enzymes for lactose. That's why we call it positive gene regulation. So negative gene regulation can be maybe a little bit harder to understand, because all we do is just like turn down the regulation. But here it's positive gene regulation because we're actually promoting gene regulation. In other words, if let's say CRP wasn't here, as long as like our repressor isn't bound to the operator, we'll make a low amount of that lactose, those lactose enzymes. But here, if glucose is scarce, the bacteria is like, oh my gosh, we need to get some energy. We really, really, really need to break down that lactose. But in normal conditions, it would have some glucose, some lactose, and lactose is more of like a, we use it whenever we, every so often, but it isn't the main energy source. Okay? Here's how you can remember that glucose and CAMP are opposites. When you go camping, what do you do around the campfire? What do you roast? Marshmallows. What do marshmallows have a lot of? Sugar. You just eat all the glucose, right? When you're done camping, you do a lot of camping, you don't got a lot of glucose lying around. I got a lot of marshmallows in my place. It's a sign I haven't gone enough camping, So then, if we go to the next slide, the opposite becomes true. Let's say now lactose is present, but now we also got a lot of glucose. So glucose is positive. That means, you got a lot of glucose lying around. That's a sign you haven't been doing enough camping in your life. So now I said bacteria want to use glucose. Because they want to use glucose, there is no camp around. Notice how CRP, because it doesn't have camp bound to it, it is inactive. So now, really key in on this. Less likely to bind. We're still making some of the lactose enzymes. Why are we still making some of the enzymes to break down lactose? Really important you make this connection here. There's kind of a lot of details. Why are we still making some of the enzymes for lactose? I just say, hey, we got glucose present. If you're a bacteria, wouldn't you want to just only use glucose? Why are they still making some of the enzymes for breaking down lactose? Thomas? Yeah, right. You still have some lactose, so your oppressor is not bonded to the operator. Okay? So we're still making low amounts of the lactose, but not as much because we aren't positively upping the gene regulation here. Okay. Yeah, that's really about everything here. So CRP, it increases the affinity of RNA polymerase. Make sure that phrase makes sense to you. Affinity means like the attraction of, or like how, in this case, like how well the RNA polymerase binds, how often it binds to this promoter and does transcription. Now CRP, the cyclic AMP receptor protein, it also will work on other catabolic pathways. So it's not just being used in just this one pathway. CRP, if you're low in, remember, CRP is active when you're low in glucose. If you're low in glucose as a bacteria, it would make sense that you would also want to activate a bunch of other genes in addition to the lactose ones. So that's why it's going to turn on other enzymes we need to break down other catabolic pathways. In other words, the bacteria is going to want to break down other fuel sources it has available to it because it doesn't have that glucose present. And again, even if glucose levels increase, as long as there is some amount of lactose present, we still will do transcription at a low rate. Okay, take a little break. Give you some time to do the mastering bio.